Hello friends, in this video we'll study about photorespiration, C4 plants and CAM plants. So first we start with photorespiration. In most plants, first organic product of carbon fixation is a C3 compound, that is 3-phosphoglyceric acid. So these plants are known as C3 plants. And what are the examples of C3 plants? They are mostly agricultural plants like rice, wheat, and soybean. Now, this all we have studied in my previous videos. What happens during very hot and dry days? The stomata of the plants they close. Then if they will close, they will produce less sugar. Why they'll produce less sugar? Because less carbon dioxide will enter and it will cause the Kelvin cycle to be less effective. Then oxygen will build up. The enzyme ribisco, as we have seen in my videos of previous videos, it will bind to oxygen now instead of carbon dioxide. Because ribisco is capable of binding with oxygen too, so it now binds with oxygen instead of carbon dioxide. Ribisco now adds oxygen to Kelvin cycle instead of carbon dioxide. Then a product is formed which splits, and then a two carbon compound is formed, because one carbon we are getting less from carbon dioxide. This two carbon compound will leave the chloroplast, it will enter mitochondria and peroxisomes. It rearranges and splits. This compound, carbon dioxide, is released after splitting. So, the release of carbon dioxide is what? It is actually respiration. Photo, because it occurs in light, it is releasing carbon dioxide and consuming oxygen. So we call this as photorespiration. Now this photorespiration has some typical features. It uses ATP and doesn't regenerate it. While a normal respiration, it will not do this. This doesn't regenerate ATP, and this feature is unlike respiration. Okay. It will produce no sugar. This is unlike photosynthesis. It is siphoning out organic material from Calvin cycle and releasing carbon dioxide that could otherwise be fixed. So, it is a wasteful process. We have an evolutionary explanation for all this thing. Previously, carbon dioxide was more in atmosphere. Rubisco enzyme has adapted now to bind to carbon dioxide more efficiently. Previously, it was more efficiently binding with oxygen. And this feature of binding with oxygen is still retained to some extent with Rubisco. So, this is the evolutionary explanation behind all this factor. But photorespiration has some protective effect too. Protective effect against the damaging products of light reactions which build up when Calvin cycle slows down due to decreased carbon dioxide. This is the only protective effect. If we can reduce this photorespiration in crop plants, we can increase their yield by around 50% and our food supplies may increase. So we should try to reduce this photorespiration. Some plants they overcome the above effect in hot dry climates by alternate modes of carbon fixations that minimize photorespiration and increase in Kelvin cycle occurs. And what are these plants? C4 plants and CAM plants. So now we'll study about these plants. First of all, C4 plants. Why they are so named? Because they have an alternate mode of carbon fixation. They'll form a 4-carbon compound firstly. And what are these plants? The example is sugarcane and corn and some members of grass family. So in hot and dry weather, these plants partially close their stomata. So water is obviously conserved and carbon dioxide is reduced in leaves. The alternate pathway used by them is started by beginning the photosynthesis in mesophyll cells and completing in bundle sheath cells. So, what are these cells? Let us have a look at the diagram. This is the structure of a cross section of a C4 plant. This is the vein. This whole thing is the vein. And around these, we see some typical cells known as bundle sheath cell. These all are bundle sheath cells. And the mesophyll cells, these are the loosely packed cells around these bundle sheath cells. They are arranged around the veins of leaf in tightly packed sheets. This is the vein, and they are scattered over here, the mesophyll cells. These are the stomata. Now, so what did we say? We said that they start by process by beginning the photosynthesis in mesophyll cells and completing in bundle sheath cells. This thing we see in this diagram when we enlarge this part of the cross section showing a mesophyll cell and a bundle sheath cell. This is the bundle sheath cell. So what actually happens? The enzyme present in mesophyll cells has higher affinity for carbon dioxide than rubisco, and it has no affinity for oxygen. So this mesophyll in this mesophyll cell, this enzyme adds carbon dioxide to phosphoenol pyruvate. Phosphoenol pyruvate forming 4-carbon oxaloacetate. 4-carbon oxaloacetate even under conditions of low carbon dioxide concentration and high oxygen concentration. Okay. Then what happens? How this oxaloacetate is formed? By addition of this carbon dioxide to PEP, then this oxaloacetate will convert to malate. After being converted to malate, which again is a 4-carbon compound, it goes to bundle sheath cells. This is the bundle sheath cell. This malate is going to bundle sheath cells via plasmodesmata. These are the plasmodesmata. Now, what is happening in the bundle sheath cells? An enzyme releases carbon dioxide, which is refixed into organic material by Rubisco and the Kelvin cycle. See here, this malate is entering this bundle sheath cell, carbon dioxide is released out of it, and pyruvate is a 3-carbon compound it is formed, which will again complete this cycle. The cycle will go on, and this carbon dioxide will enter the Kelvin cycle. Okay, this is what is happening. Simultaneously, pyruvate is regenerated, which goes to mesophyll cells. As I told you, this pyruvate is going to the mesophyll cell. Then pyruvate is converted to PEP, and again C4 cycle in mesophyll cell continues. This C4 cycle continues in the mesophyll cell. This procedure requires ATP. This whole procedure is requiring ATP, so we can say that it is a price of concentrating the carbon dioxide in bundle sheath cells. The carbon dioxide is being concentrated in this bundle sheath cell, and ATP requirement is the price which the plant has to pay. And this ATP is used for cyclic electron flow. Okay. These plants have only PS1 and no PS2. Photosystem 2 is not found in these plants. So they have only cyclic mode of electron flow. And the non-cyclic flow, as we saw in our previous videos in light reactions, is not found in these C4 plants. So actually, carbon dioxide is being pumped. This carbon dioxide is being pumped here from mesophyll cells to bundle sheath cells. So carbon dioxide concentrations remains high in bundle sheath cells. This is what we want. So Rubisco will now easily bind with carbon dioxide instead of oxygen. Or we can say that an ATP powered pump is sending carbon dioxide to that place where fixation can occur. This is how the C4 plants, they are avoiding photorespiration and they are using the carbon dioxide by pumping it into the bundle sheath cells at the cost of this ATP pump. C4 plants thus thrive better in hot, arid environments. As we are facing the problem of global warming, although C3 plants are benefited by high carbon dioxide in atmosphere because we are living in a highly polluted environment where we have high carbon dioxide, and C3 plants, they work better with high carbon dioxide, but they go to photorespiration in high temperature. If the temperature, when we are facing the problem of global warming, we have high temperature and the pollution, it will give high carbon dioxide. So C3 plants, of course, they can work better in high carbon dioxide, but not with high temperature. And C4 plants are unaffected by oxygen or carbon dioxide concentration or temperature. So they are better adapted in today's era and can meet more food demands in coming future. Okay, I hope you have understood the C4 plants functioning. Now, what are CAM plants? Coming to CAM plants, the example of CAM plants is pineapple and some cacti. What they are doing, they open stomata at night and close during day. This is how they conserve water. But carbon dioxide entry is also prevented. So what they do, they take up carbon dioxide at night and convert it into a variety of organic acids. So this is their way of carbon fixation. Crassulacean acid metabolism or CAM metabolism is the name given to them after to a succulent family of the same name. These organic acids are stored until morning. So they will do all this thing at night because the stomata are open at night, and they'll store these acids so formed till morning. And during day, via light reactions, ATP and NADPH are supplied for Calvin cycle. At that time, carbon dioxide is released from organic acids, hence forming sugars. So let us have a look at the diagram. This diagram is showing the functioning of C4 plants and this is showing the functioning of CAM plants. See what the C4 plants are doing via mesophyll and bundle sheath cells. They are taking carbon dioxide in mesophyll cells and pumping it into bundle sheath cells, and they go through Calvin cycle and finally produce sugar. The same thing the CAM plants, they are doing at night, taking carbon dioxide, fixing it, converting to organic acids, and in day releasing this carbon dioxide, and Calvin cycle goes on and sugars are produced. So these mesophyll and bundle sheath cells are not like this. The same thing they are doing in night and day. CAM, C4 and C3 plants all are finally using Calvin cycle. Don't forget, Calvin cycle is occurring in all these three plants to make sugars from carbon dioxide. So the ultimate way of producing sugar is Calvin cycle only. So, this was all about photorespiration, C4 plants, and CAM plants. Thank you for watching the video.